Hi class, and welcome to your next video on meiosis. Now meiosis is going to help us create a new type of cell. In mitosis we created body cells, or what we call somatic cells. Now we're going to be creating our reproductive cells that we are going to be using for sexual reproduction. So gametes have a single set of chromosomes. Now your sex cells are considered gametes. Any cell with two homologous sets of chromosomes is called a diploid cell. So that's anything that is in your body that is not a reproductive cell. So we abbreviate that 2N. And in humans, 2N equals 46. So we have 46 chromosomes in our diploid cells. So our body cells, or somatic cells, are all diploid. So any cell besides an egg or a sperm is a somatic cell. The only cells that are not diploid are our gametes, our sex cells, egg and sperm cells. So each gamete has a single set of chromosomes. You have 22 what are called autosomes and one sex chromosome, either an X or a Y. These are our haploid cells. They have half the amount of chromosomes, and it's abbreviated N, and N equals 23 for 23 chromosomes. So the reason we have half the amount of DNA is because when we join that egg and sperm together, that we reunite them so that we get those 46 chromosomes. So if we look at the human life cycle, we can see that we go through periods of diploid and haploid in our cells to create a diploid cell. So if we start up here, our egg and sperm cells are haploid. We go through fertilization, which is going to create a diploid cell. We're going to go through mitosis, which is going to create identical cells, and they're going to all be diploid. And then we're going to go through meiosis as adults to create haploid cells to create and reproduce more life. So moving forward from that, becoming diploid. To become diploid from that haploid state, what you need to do is you need to go through fertilization. So the process of the ovum and the sperm being united in sexual reproduction. This results in what we call a zygote, and that is diploid due to the joining of the mother and the father's DNA. The life cycles involve alternation between the diploid and the haploid stages, and haploid stages are made in the division that is called meiosis. Okay, so meiosis reduces the number of chromosomes from diploid to haploid and is a type of cell division that produces haploid gametes in the diploid chromosomes. If we look here, remember interphase from mitosis. We're going through the same thing here. Interphase is going to duplicate our DNA. It's going to grow our cell, and it's going to get it ready for meiosis. In meiosis 1, we're going to split into two cells, but we're going to become haploid in meiosis 1, and then we're going to split it even further in meiosis 2. So interphase is going to be exactly the same as it was in mitosis with the cell cycle. The DNA is going to duplicate. We're still going to start with chromatin. The nuclear envelope is still going to be intact. We're going to use centrioles still. And the cell is going to grow in G1 and G2. In prophase 1, which is our most complex phase during meiosis, first we're going to have synapsis. Synapsis means our chromosomes are going to come together and form what are called tetrads. When they come together, what's going to happen is the homologous chromosomes that are the same size are going to match up with each other. And that's going to form the tetrad. So chromatids are then going to exchange segments of DNA in a process called crossing over. And what happens here, as you can see, the adjacent chromosomes are going to cross over, and they're going to form what's called a chiasma, and they're going to actually exchange that DNA And so this is going to help with our genetic variability because now we have pieces of one chromosome on another chromosome that's going to go into different cells. So it's going to help provide for genetic variability. This is one of the reasons you do not look like your siblings. You are not the exact replica of mom and dad's DNA, but everyone is unique because of this crossing over. So the next thing that's going to happen is metaphase 1. The chromosome tetrads move to the metaphase plate. So they're not going to line up like they did in mitosis. What they're going to do is you're going to see the actual tetrads and the homologous chromosome pairs are going to be lined up together. And that's going to provide for what's going to happen in anaphase 1. In anaphase 1, we are splitting, but the sister chromatids are going to remain intact and they're going to remain attached to their centromere. We're not pulling them apart just yet. We're going to wait until meiosis 2 to actually pull them apart. So we're going to leave them intact, and we're going to use this to help us form our haploid cell. So in telophase 1, chromosomes arrive at the poles. Each has a haploid chromosome set in duplicate form. So we still have the duplicates. We have to split it one more time, but we only have half the DNA because we had put it into the actual homologs and tetrads and split it that way instead of lining everything up in the middle. This is the difference between meiosis and mitosis. So we now have haploid daughter cells, but we need to split it even further so we don't have the duplicated form. So in meiosis 2, we're going to go through prophase 2, metaphase 2, anaphase 2, and telophase 2. These are very, very similar to mitosis. In prophase 2, your spindle fibers form and move chromosomes toward the middle of the cell. In metaphase 2, chromosomes are aligned in the middle of the cell. In anaphase 2, the centromeres of sister chromatids are pulled apart. So we're actually going to see those pulled apart, and they're going to be pulled towards the poles of the cell. In telophase 2, the nuclei form at the cell poles, and cytokinesis is going to take place. So let's look at this. So we go from the cleavage furrow here where we have the haploid cells to, again, going through prophase 2, metaphase 2, and it's almost identical to mitosis now, except we are starting with a haploid cell, so we must end with a haploid cell. And at the end, we're going to have 4 genetically different daughter cells. Instead of, like in mitosis, you have 2 identical daughter cells. So we have our genetically different daughter cells. So using this, we have 3 different ways we're going to have genetic variability. So we have the law of independent assortment. It's just the ability of different chromosomes to assort independently and differently into different gametes. We can have any combination that meiosis provides for. There's no rhyme or reason to why a chromosome is going to go into a certain gamete. There is no set assignment. There is nothing. So it can assort independently. It can assort on its own. The next part of our genetic variability is random fertilization. There is nothing that says one sperm has to fertilize one egg. Any sperm can fertilize any egg. It just has to be of the same species. So it has nothing to do with what is in that sperm or what that sperm contains. It's just that it is the sperm of that species to fertilize that egg. And lastly, what we already talked about in prophase 1 is crossing over. When we break off parts of that chromosome, we are adding a little bit of variability to what is in that gamete, and we're giving it more uniqueness and variability. So, now let's think about this. Accidents happen all the time. They happen in biology too. And we know accidents happen because we know about things like Down syndrome. So, accidents that can alter our chromosome number are going to be mistakes that occur through meiosis when our chromosomes do not assort correctly. And this is called non-disjunction. Non-disjunction is when members of chromosomes fail to assort, chromosome pairs fail to separate correctly. So what we see here is this is truly non-disjunction here. These guys did not separate. So what's going to happen is one of our gametes is going to have 3 of the chromosomes and one's going to have 1. So one of our gametes might get an extra chromosome where one gets one less chromosome, which results in these conditions like Down syndrome and Turner syndrome. Extra chromosomes generally do not result in death. Extra chromosomes just result in difficulty, like Down syndrome. So in order to diagnose these disorders, though, from non-disjunction, a karyotype must be used. A karyotype is an ordered display of magnified images of an individual's chromosomes arranging in pairs starting with the longest. So as you can see, we start with number 1, we go all the way to 22. They get smaller and smaller and smaller, and they hold less and less DNA. And then we get to the sex chromosomes, which are the X and the Y. And for a male, it's an XY. For a female, it's an XX. So as we can see, this karyotype we are looking at is of a male. And the Y actually does not carry that much information as opposed to the X. If you look at the size, they're much different than like the 11s and the 12s and the 18s. They're very similar in size. But the karyotype is just used for diagnosis and to look at those chromosomes. Okay, this is all we have for today, so make sure you took great notes. Make sure you got lots of diagrams into your notes. There's going to be a lot of diagrams in this chapter. Make sure you know these diagrams. And also make sure you are ready to talk about this. And we will be talking about this after break.